Welcome. This explainer is designed to provide a high-yield, mechanistic overview of oncogenes, tailored specifically for your CSIR-NET preparation. We'll focus strictly on the core concepts you need to know.
Here's our plan. We'll start with the fundamental definitions, then move into the normal function of proto-oncogenes. After that, we'll classify them functionally and walk through the key examples and cancer associations that frequently appear on the exam.
All right, let's begin with the core definition. What exactly is an oncogene?
An oncogene is, simply put, an altered version of a normal gene. Its key feature is that it functions autonomously. This means it can drive cell growth and division all on its own, without needing the usual external signals. This uncontrolled promotion of growth is a major contributor to cancer development.
Now, this is a critical concept you have to grasp. The mutations that create oncogenes are gain-of-function mutations. What does that mean? It means the mutation gives the protein a new or enhanced activity. It's a dominant mechanism, so mutation in just one of the two alleles is enough to produce the oncogenic effect.
So to really understand oncogenes, you first have to understand where they come from. And that brings us to their normal, unmutated counterparts, the proto-oncogenes.
Proto-oncogenes are completely normal and, in fact, absolutely essential genes. Every healthy cell has them. They encode the proteins that regulate cell growth, division, and differentiation in a very controlled, orderly way. They are the responsible managers of cell proliferation.
This comparison really gets to the heart of the matter. You see the shift from a normal gene to a mutated one. But the most important line here is the second one. The function goes from being regulated to being constitutively active. That just means it's always on. And that, of course, is what leads from controlled growth to uncontrolled growth.
This diagram shows you a typical signaling pathway that controls cell growth. It starts with an external growth factor, which binds to a receptor. This kicks off a cascade of signals inside the cell, which eventually reaches the nucleus and activates transcription factors. Every protein in this chain is a product of a proto-oncogene, and each one is a potential target for an oncogenic mutation.
Okay, so now that we've seen the normal pathway, we can classify oncogenes based on where their protein products fit into this scheme.
We can break the pathway down into these four key stages. You have the external signals, the receptors that receive them, the transducers that relay the message inside the cell, and finally, the transcription factors in the nucleus that carry out the orders. An oncogenic mutation can happen at any of these levels.
Let's start at the very beginning of the pathway, with the growth factors and their receptors.
In a normal scenario, cells wait for growth factor signals from other cells. But some cancer cells figure out how to make their own. This is called an autocrine loop. The cell stimulates itself. A classic example you should know for the exam is the overexpression of PDGB, which comes from the C-cis proto-oncogene seen in astrocytoma.
Now for the receptors. They become oncogenic when they get stuck in the on position, sending growth signals even when no growth factor is present. There are two main mechanisms you need to remember. First, point mutations can lock the receptor in its active shape, like with EGFR in lung adenocarcinoma. Second, gene amplification can lead to way too many receptors on the cell surface, which is the case for HER2 in breast cancer.
All right, let's move inside the cell and look at the proteins responsible for relaying the signal from the receptor down to the nucleus. This is a very diverse group of proteins. You have GTpases like ROS, which act as on-off switches, and you have various kinases like ABL and BRAF, which pass the signal along by adding phosphate groups to other proteins. The common theme for them as oncoproteins is that they all get locked in a permanently on state.
Okay, the RAS family is extremely important. You must know this. These proteins are active when they're bound to GTP and inactive when bound to GDP. Oncogenic mutations destroy their ability to turn themselves off. So RAS gets trapped in the active GTP-bound state and just relentlessly activates downstream growth pathways.
So where does this signal cascade end up? It ends in the nucleus with the transcription factors. These are the proteins that bind to DNA and turn on the genes needed for the cell to divide. An oncogenic transcription factor, like MEK, is usually overexpressed. It then forces the expression of genes that push the cell through the cell cycle over and over again.
All right, to finish up, let's consolidate everything into a high-yield summary of the key oncogenes and the cancers they're associated with. This is pure exam recall material.
Take a good look at this table. These are the associations you are very likely to be tested on. Notice the receptors, EGFR in lung cancer, HER2 in breast cancer. For the signal transducers, KRAS is huge in pancreatic, colon, and lung cancers. ABL is famous for the translocation that causes chronic myeloid leukemia, and BRAF is key in melanoma. And finally, the CYC translocation is the hallmark of Burkitt's lymphoma.
And one final, absolutely critical takeaway. Remember this: the RAS family of genes is the most frequently mutated class of oncogenes found in human tumors. It plays a role in a huge percentage of all cancers.
As you continue your studies, reflect on this question. It's not just about which gene is mutated, but also how it's activated. Understanding that distinction is key to a much deeper comprehension of cancer biology.